Okay, hello. Welcome to today's Cambridge Rare Disease Society's talk featuring Professor David Rowitch, who is a developmental neuroscientist and head of the Department of Paediatrics here in the University of Cambridge. His laboratory in the Wellcome MRC Cambridge Stem Cell Institute investigates genetic factors that determine diversity of glia, which compose 90% of the cells of the human brain. And he's an academic lead for the new Cambridge Children's Hospital, which will incorporate genetic medicine to augment routine pediatric practice. And today he will be covering advances in rare neurogenetic diagnosis in the NICU and new treatments for Pelizaeus Merzbacher Disease, PMD. And so without further ado, let me introduce Professor Rowitch.
Thanks very much, Jenny. So I guess as Jenny said, I'm going to cover a range of conditions. And all of these are obviously pictures of patients. And by the end of the lecture, you'll certainly know what conditions they have and whether you might order a genetic test for them. Closures. And what I want to cover will be an approach to genomics. I'll tell you what the neuro-NICU is. I'm a neonatologist. And how genomic medicine is progressing in the UK, and why that's actually especially important in pediatrics. I'm going to tell you about a cell called the oligodendrocyte, which is the myelinating cell of the brain. There will be pathways, so you're forewarned. There's going to be some basic science here, but that helps us understand the nature of problems going on in humans. And then how can we use that information to translate to precision therapies, gene therapies, cell-based therapies, which in the future some of these patients will actually be eligible for some now. So here's I'm going to start with the case history. You can sort of read it yourself, but it sort of suggests here's a baby is born with some difficulty, unable to breathe, blue. So this is something you might have in your pediatric rotation. You're called to the delivery room, have to assess this baby. And so respiratory support is initiated, and then the baby starts to breathe on his own. They assign APGARs. We'll see why in a second. And then as you look at the sort of history as the child grows older, has further more difficulties. Unable to walk, starts to develop spasticity or contractures. Movements become uncoordinated, develops seizures, and has little speech or able to feed. And so is this a case that could have come from Rosie Hospital? Yes. This could happen. This could happen from the home birth. You might be called emergently because of a situation like this. But in fact, this is part of the original case series of William John Little from the 1860s that became to be known as cerebral palsy, originally called Little's disease. This paper describes 58 horrible cases of, bad things happening around time of delivery, resulting in a child who grew up to have neurological disabilities. And so it really started this idea that traumatic birth situations could cause injuries leading to long-term neurocognitive developmental problems. So, but the question I'm going to ask here is, would this child benefit from genome sequencing? Okay, so cerebral palsy, tens of millions of people worldwide. This is probably an underestimate. Many of the kids that are subject to these brain injuries will die, sadly, in the first year of life. Some of these are caused by genetic mutations. You'll know the answer to that sort of by the end of the lecture. Then in NICU, you'd also have preterm babies, maybe 10,000 babies per year that are preterm. And they're vulnerable to neurological damage and also can have mental health problems later on. So as we start to follow these cohorts into adolescence, we actually see that the brain is affected in other ways. And seizures, which is the most common reason that most common neurological diagnosis requiring admission to NICU, 2,000 people. Okay, so let's talk about an approach to seizures. Now, as a neonatologist, this is a place where you can start to think about stratified medicine. It is the most common neurological emergency, so it happens a lot. Challenging to diagnose. It has a varied prognosis because it depends on the cause of the seizures. You can kind of appreciate why that might be the case. But the neonatologist is a protocol-driven physician. We like to resuscitate, we like to put tubes in things, babies, and then when it's a seizure, we give one medicine. And in the US, it's phenobarbital. Here it will be, Keppra or phenobarbital. In Europe, all kids get the same thing. And you can ask yourself, is that the appropriate thing to do. One size fits all might not be the right way to do it. So at UCSF, we started something called the Neuro Intensive Care Nursery, and that was to develop a stratified approach so that we could sort of kind of get it right from the beginning. And this is a multidisciplinary unit that would involve nurse practitioners, specialized nurses that are, focused on babies at risk for neurological injury. Lots of monitors around the bedside, so ventilator, but also EEG and other monitors. Hypothermia, which is now a kind of a therapy for hypoxic ischemic encephalopathy. So this baby, which you can barely see, is actually under a blue blanket. That's a cooling blanket. This baby will be maintained at 33.5 degrees centigrade for 72 hours. And that provides a lot of brain protection. So a multidisciplinary effort to optimize that care, including neuroimaging. Now, Top and Austin here in Cambridge decided to replicate that model and develop the Rosie NICU. And so you can see examples of how this works here in Cambridge. But in the NHS, there's a regionalized approach so that we can now start to think about the scaling of these problems. And there are about 240 babies with neonatal seizures per year. Now there's a uniform seizure protocol that goes across the region, and now you can start to use, this stratified approach. And also, lots of deliveries per year. So for all neonatal conditions, the scale in the NHS really gives you some ability to answer questions that you couldn't answer in a place like San Francisco. So here's a stratified approach. Now this is actually a video of a little baby, and it's running, believe it or not. So I could ask you the question: is this baby having a seizure? Now, is there anything that makes you think this baby's having a seizure? It looks like that in the video. Looks like he's sleeping. You can see the stat monitor in the upper right, and seizures can cause hypoxia. So, but that stat is not 100%, it's 8%. And another kind of clue here that blue blanket is the baby being treated for hypoxic ischemic encephalopathy, high risk for seizure. But the thing about neonatal seizures is there's a clinical 50% of the time. So here's the EEG of this baby, and status epilepticus. So for the neonate, unlike other epileptic kind of scenarios, you can have 50% of seizures are subclinical, which means you have to do continuous monitoring because there's no other way to detect the seizure. So EEG becomes part of the stratified approach. MRI becomes part of the stratified approach, and you can do it in a very clever way where here's baby in an incubator with the MRI coil inside the incubator. So now you have improved resolution of the MRI. It's quieter, baby's nice and warm, can withstand even an hour-long research MRI sequence. And that's a piece of kit that we also have. And now we can say, well, does this baby have a stroke? We should pick up by MRI, or hypoxic ischemic encephalopathy. The MRI helps you with that, or brain malformation, because that could also cause seizure. So MRI is important. We screen for metabolic conditions or infection. But then here's the thing that really is an asset in the UK is genomics, because there are rare genetic conditions that can also cause these epilepsy syndromes. And if you are having a genetic diagnosis, genomics is the only way to really tell that. So when I came to Cambridge, I started working with geneticists Lucy Raymond and our postdoc Courtney, where we just wanted to kind of look at how useful whole genome sequencing was to diagnose conditions in NICU, including seizures. Now, whole genome sequencing is the most comprehensive DNA test. There are, of course, many others that you can use. And this slide lists the number of conditions that you can pick up by whole genome sequencing. And some of these you can't pick up by whole exome sequencing. For example, mitochondrial disorders don't usually show up on exomes. Structural variants and copy number variants not very good by exome. So you get more bang for the buck. And in the UK, fortunately, whole genome sequencing is more and more available. So that was our question. I think the next slide will be a video kind of, here's the other thing about whole genome sequencing, quick. In fact, we're starting to pilot 24-hour turnaround whole genome sequencing for pediatric cancer in Adbrooks. The only place in the world that's doing that for pediatric cancer. So now, it really could become a tool that gives you fast results in NICU or ICU. That's great. But the next is a video from a couple of years ago, which is kind of describing the impact of whole genome diagnosis with the families.
This advice and its most vulnerable, made even more Billy May and her parents, Claire and Chris, each had their genome, their entire genetic code sequenced to try to discover why she was having life-threatening seizures. The results showed Lily May has a rare form of epilepsy caused by a single gene error not inherited from her parents. It led to immediate improvements in her care.
Since we've had the diagnosis, it's been a lot better. We had to change one of the medications due to the fact that the one that she was on was obviously aggravating the type of epilepsy that she has. We saw a big difference as soon as that change was made.
This process, that one test result actually allowed us to put all the correct people in place and make less for her.
Sequencing the billions of letters of DNA code that make up a genome used to be hugely expensive. Now it costs less than £1,000. Scientists in Cambridge analysed the genomes of 350 children in intensive care, comparing it to the DNA of their parents. They found 1 in 4 children had a genetic disorder and were able to give the diagnosis in 2 weeks. This study shows conclusively that whole genome sequencing is of real benefit to patients, speeding up diagnosis and helping to find the right treatment. From next year, throughout England, the NHS will offer whole genome testing to all babies and children where the cause of their illness is unknown. The first national health service in the world to do so.
So that's a kind of nice venue. It gives you some sort of sense of precision medicine from parents' point of view. And actually, Fergus was right that the NHS actually, as a result of the study, did pick up for NICU and PICU, the R14 pathway was kind of the result of now embedding this in the NHS. That's an exome, rapid exome, but now it's going to a rapid genome. So now we can start to think about how useful are these tools. So this Next Generation Children's project included 521 patients and their parents, so we call those trios, and then you can kind of see, like, okay, where's the clinical units where you get the most bang for the buck, and what are the conditions that you might, sort of really want to get a genome. So here's Nikki, where they covered this, 194 patients, 25% diagnostic rate. That's pretty high. You might think if you're an intensivist, you might want to order everything. Or in pediatric ICU, 31%, 1 in 3. And then there's some diagnoses like hypotonia, floppy baby. And the way we manage floppy baby is essentially we wait to see if that stops being floppy. That's the medical approach. And then if the baby doesn't stop being floppy, then we start to do tests, including muscle biopsies and all kinds of basic things. It takes 6 weeks to diagnose a hypotonic. But now we go to genome sequencing because it's 50% have a genetic condition. And that will help us, think about medications or other approaches that would be better, or just save them from the invasive procedure. We won't do the muscle biopsy because we know the answer from this test. PICU, now we will use this routinely. Pediatric neurology, 1 in 2 patients. So every child coming in the door for pediatric neurology should have a genome, just because the a priori risk is so high. And now we're going to start making more diagnoses, and then this sort of summarizes what that study showed and why it kind of was impactful. These are all NHS patients, and it has led to changes in care across the country. So now we can order the whole genome for pediatric cancer and for NICU and PICU. For neurological conditions, it's also on the test record. Unfortunately, it doesn't take 2 weeks. It takes about a year. So the turnaround time is the thing that we knew to work on is getting faster. But you can say that once we have a diagnosis, you mitigate that diagnostic odyssey. That's that months or years without a diagnosis. You can answer that with the genome. And you can change care more and more. We're learning how to stratify patients and then see how they can respond to different drugs. So Millie May, who has an SCN1 mutation, actually was one of the anticonvulsants to avoid. It was aggravating her condition, so she could be switched to other things that work better for her. And if you have, conversely, KCNQ2 deficiency, you can have status like that baby that I showed you with EEG, in really, really difficult seizures to treat. Then you put them on carbamazepine, and it's like this totally different thing. Baby will wake up, start breastfeeding, goes home with mom. It's really remarkable how this can work. And so we're learning more and more. Maybe we take great industry to start cataloging this. There are a number of others. So now we can go back and look at our diagnoses we made in our project. There were 59 babies that had neurogenic seizure syndromes, and the ones that are highlighted in turquoise are the ones that are eligible for precision medicine, either anticonvulsant drugs or now tritabolone, which is more around mitochondrial disorders. It's about 1 in 5 kids with these, and that's just going to go up. So we're going to get better at doing repurposing based on these genetic diagnoses. So we could say gene discovery, precision medicine, it's going to take us, further and get kids on more appropriate drugs. But big questions in many conditions that happen with treatments, and it won't just be a matter of repurposing. So we do need to go into the lab. We do need to think about models of disease and kind of discovery. So that's where I want to talk about another disease with a name that nobody would probably want to say 10 times as quickly, Pelizaeus Merzbacher Disease, or PMD. And this is a child, when born, had stridor so significant, had to have a trachea put in, and it was a child with AJ, wheelchair bound, and this is the severe neurodevelopmental impact of Pelizaeus Merzbacher disease. So what's going on with that disease? And for that, you have to understand the myelinating cells of the brain, which are called oligodendrocytes. So in our brain, of course, we have neuronal impulses that are being transmitted, and you've learned that in your biology course. You may or may not have learned about the white blobs on the nerves, which is myelin segments. And this is what actually massively increases the speed and reduces the diameter of axons, so you can have more axons and they fire much more efficiently. So myelin is made by oligodendrocytes in the white matter of the brain, and these are the cells that are affected in Pelizaeus Merzbacher disease. We can measure myelin by various techniques using MRI. So we've got a non-invasive technique to be able to say, is there enough myelin in the brain, etc. One of the lab's focuses is on the oligodendrocyte and how they develop. And so my lab discovered genes called OLIG1 and OLIG2, and they are transcription factors that function in a multipotent neural stem cell. And they're able to give rise to neurons or oligodendrocytes or neurons. But if you want to make a oligodendrocyte, myelinated cell, you have to have OLIG2. And that enables you to get to this stage where you can make this oligodendrocyte yourself. They are supercritical cells. I won't have a lot of time to talk about them, but they are proliferative and migratory. Imagine, like you had just 1 or 2 of them. They will eventually fill up the entire central nervous system. They're just colonizers. They look for axons. They want to myelinate them to sort of provide that support. And when they find axon tracks, they're called pre-myelinated oligodendrocytes, and then they will engage with an axon and form that myelinated structure. Now that looks like a fairly simple this to that, but what that cell is actually becoming something like this. So somewhere in the middle of that is a oligodendrocyte, and it's setting off hundreds of different arms, and it's coding multiple axons all over the place. And so this is a increase in cell surface area of 7,000-fold. So the oligodendrocyte is the largest cell in the body. And imagine the metabolic stress of having to make all of that proteolipid to code all these, it's just a really remarkable cell. And how, so then you can also start to appreciate that this transition, because it's so metabolically active, is a checkpoint in a number of diseases. So I'm not going to talk about multiple sclerosis, but in MS, you can find these cells that are trying to repair a demyelinated MS plaque, but they're blocked. Probably because the cell is thinking, this is not the right environment to become that cell. And so they have checkpoints that stop them. And you'll see that it's a checkpoint also with Pelizaeus Merzbacher disease. It's a real crisis. Things have to go just right. If you have a genetic problem, if you have an environmental problem, you may not be able to make that transition. So that's a bit of background on the oligodendrocyte, how they're relevant. So here's what I was telling you about the MRI being where you can measure PMD and its white matter effects. Then on the upper left is a T2-weighted MRI at 2 years of age. And in this particular T2, the appearance of myelin will be charcoal. So the turquoise area is pointing to, kind of like some myelinated tracks going to the anterior posterior axis, and then compare that to the PMD on the right. Now you can detect myelin at age 2. The PMD patient has opacity where it should be charcoal color, and that would represent diffuse hypomyelination. And they have a mutation of a myelin protein called protein 1 or PLP. And different kinds of mutations can cause a mutation or a duplication, like more of the gene is bad, or null mutation, none of the gene is bad. It's a very kind of tricky one. It has to be just kind of like in its normal, natural state. Perturbations really cause problems. So you could have, let's say, one amino acid change in one part of this protein, and that gives you severe PMD. And these boys will fail to sit. And it is excellent for sepsis. These textbooks. Boys fail to sit up, crawl, top. Death is usually by 10 or 20 years to maximum support. When we started the studies on this, clinical studies on this, there were no therapeutic options. Again, it's a disease of oligodendrocytes, so we're trying to also bring the perspective of our biology to understand like why it is so bad. You know, this little boy actually has this mutation, and this is supposed to be a glycine, but in this little boy it changes to a glutamic acid. So now you have an acidic amino acid right in the middle of transmembrane spanning domain in the lipid bilayer, doesn't like it, or you know, you negative charge. Acidic amino acid is hydrophilic and hydrophobic. You don't want it there, and so what does it do? And then you know, with these sort of deep mind kind of modeling things, you can actually kind of come up with theoretical models of the PLP protein in the wild type, and you can look at it over time and say, well, this would be the full structure of the membrane of the wild type. And then after 460 nanoseconds, this is what it would look like. Membrane is not too bad. But here is now that mutant form at 0 and 460 seconds. You can see how the membrane is now being perturbed. And if you imagine that that's happening on the scale, now the cell is probably being put under tremendous stress. And that could be how a point mutation like that could cause destruction of the normal structure. Again, I'll come back to that to share some further evidence. But here's how you can let's start to think about these PMD patients. So they've got a problem. There are oligodendrocytes. When it gets to the point that they're making myelin, it makes, you know, these mutant proteins that are starting disrupting the cell biology. They're not making myelin. You see diffuse hypomyelination. They have what you could say are defective oligodendrocytes. So the logic is, could you replace them with cancer work, or could you repair them? And then measure outcomes by clinically and by. So there are three ways you can think about, you know, doing this. One would be to fix the mutant PLP gene. One would be to find a drug that helps the cells work better despite the mutation. And the third is that you can replace them with transplant of you know, functional oligodendrocytes. Actually, all three of these are being tried for PMD. So it's a nice example of like how could you approach it and how you come up with some research solutions. Okay, but again, it's line result. What's a neural stem cell? Let me talk about that approach first. Neural stem cell is a multipotent self-renewing cell. You give rise to cardinal lineages of neurons, oligos, and astrocytes. For PMD, we only care about oligos. So the study that I did in CSF was to take multipotent human neural stem cells that came from a donor brain, and then implanted them into white matter of 4 boys that had Pelizaeus Merzbacher disease. And then the idea was to be able to see if they were safe. This was a phase 1 study. So phase 1 studies, they're usually small, they're only powered to say, is this a terrible thing to do because it's going to cause a horrible complication and you never want to do it. That's kind of the answer you're trying to get. But you can also try to build in some efficacy measures. You don't expect to see clinical improvements, but you might want to see these MRI changes. So what we did was to do these implants, and then we would do serial MRIs and then measure the area that was grafted versus regions of interest, we so-called, that were not engrafted. So blue ones didn't get cells, the red ones did get cells. And then they had MRIs at 0, 3, 6, 9, 12 months, 2 years, 4 years. We followed them all the way to 5 years to kind of look at changes. And what we can see is in some of these areas where the cells have been grown, you would see MRI changes that were characteristic of myelin. So something called fractional anisotropy, a positive inflection is associated with myelination. So we would see in a region of interest increasing FA, suggesting the possibility, those cells have gotten in there and engrafted and myelinated. And relative radial diffusivity is another metric you can associate with myelinated axons. It goes down, the more myelinated. And so you see that also reflection that would suggest that possibility of myelination taking place in those areas. At 4 years of age, in 2 of the 4 boys, we saw, and in this case, these are T1 flare, and that's the myelin appears sort of like a white hyperintensity, you could say. So what the arrows are pointing to are areas that look to the radiologist like they've been myelinated. They have increased FA, they have decreased radial diffusivity, they have flare T1 hyperintensity. Looks positive. To be convinced, you have to do a biopsy. We're not going to go back and do that because that would be ethical. But it was suggestive of myelination taking place. So phase 1 study, what we could do is say, getting the cells in, was that safe? The immunosuppression thing beyond, is that safe? Were the cells safe? We concluded yes for all of those at 5 years after implant. That there were some MRI findings that suggested engraftment, but we couldn't say this is therapy yet because we have no histological evidence that they're myelin. We can't determine efficacy on the small study. You need a phase 2 or phase 3 to determine that. And in this particular case, the sponsor company broke and was not able to do, so we couldn't do the phase 2 study. But it's an illustration of, at least it was positive foundational study to kind of say, okay, others could maybe take this forward. And there is another company doing a very similar study design to ours with a phase 2, phase 3 design. So we'll get an answer. But again, there's a path forward to think about cell-based therapies for conditions of the brain, in particular PMD. Okay, how about finding a drug that helps their cells work better? Now, this was actually a study that was requested by the parents. So the parents found out that, oh, well, there's things called iPS cells. And so can't you kind of start to model PMD from our kids and see if you can figure out something that would help them? And so we did this study with Marius Bernig at Stanford and my postdoc at Rocco. And so you know how to make an iPS cell. You can take a skin cell or other cells. You can convert them into sort of pluripotent cells, sort of pushing them back in time. And then from these iPS cells, what you can do if you have a genetic kind of disease is you can also do a gene correction. So that gene correction is really helpful because you have, let's say, a mutant PMD cell, and now we gene correct the PLD mutation, and now that will probably behave like a normal cell. Now you can compare one to the other, and it's the right genetic background for comparison because it's the patient's cells. You've only corrected that gene. And now you can differentiate them into different cells like cardiac cells or oligodendrocytes. And then you could then do drug screening and see, could you find a drug that helps them so better? So that was the approach that we took. And then to show you some of the data, both from animal models and from cell culture, here are a couple of markers to keep in mind. One is called O4, and that's pre-myelinating marker. Another one is called Ermin or MVP, and that will be for the mature cells. So, and then this question is this a stress point? So maybe the PMD cells never make it there because they're stressed out, or so that's what we consider a test. Now, one of the stress pathways that you will again probably learned about in biochemistry is ER stress pathways, also called the unfolded protein response, and that's one of the ways that cells, unfortunately, commit suicide in the face of overwhelming stress. So you can have ER stress that could come from reactive oxygen species or chemo or radio insults, things that could generate reactive oxygen species and will activate the PERK pathway, factor called PIRF2 alpha, and then you can get upregulation of another factor called HF4 and CHOP. And if these two things are up, that's bad. That means you're going to become a dead cell through the apoptotic pathway. And that's was one of the pathways that we considered could have been a problem with PMD. So in this case now we created a mouse which is reflecting that exact mutation in the patient that I showed you before. That transmembrane spanning domain 2 mutation, point mutation, change that neutral amino acid to an acidic amino acid. And now we can see what happens in the mouse to that kind of cell. And on the top is the unmutant one. The bottom one will actually have the mutant. And the top, you can see that red marker ermine, that's the mature cell. There are lots of them. So, the wild type or the, unmutated has no problem. You can already appreciate there are very few ermine cells here. That means they're not making it to that mature final stage. And what we can also see in the mutant is they start to turn on CHOP, and they start to turn on the KIP1. And those are the signature cell pathways. So essentially what happens in this mouse, we can say, is they get to the point where they're just about to myelinate. They probably start to make the mutant protein. The mutant protein distorts the membrane, and it causes stress leading to apoptotic cell. Now that's actually an insight about how PMD occurs, because we knew there was hypomyelination, but we didn't know why. In this case, we're going to say the reason that there is no myelination is the cells to make the myelin are gone. So now you can sort of see what we're fighting against. In the iPS cells, essentially we found the same thing, where these are now the mutant oligodendrocyte. These are this comparison of mutant oligodendrocytes and those that have the gene correction. And then we're measuring them with the OFA marker, immature cell, and then the MVP marker, the mature one. Okay. So they can both make real 4 cells. That's the pre-myelinated cell. But look what happens when you get to the MVP positive stage. The gene corrected are just fine. The mutant ones don't look too happy. And actually, these ones are starting to undergo apoptosis. They're coming to double. So we saw very few of the MVP positive or mature cells in the PLP. So again, just like in the mouse, they just can't make it past that stress. But maybe we can make it better. The other thing we could do is use transcriptomics to figure out what's going on in these cells that are trying to make myelin. We found that there was iron metabolism genes that were highly dysregulated, and that gave us an idea to drive the trial for helping those cells. And that would be an iron chelator. Now, iron chelators are things we use clinically for human chromatosis, for people that get a lot of transfusions, beta thalassemia. They will be on iron chelators because they get iron overload syndrome. So that's actually a drug that's on the chest. So let's now look at our mutant oligodendrocytes in culture compared to the gene corrected. They don't look too happy. These are happy with big bushy sort of like octopus cells. But now in panel E, they've got the iron chelator. So this has the genetic problem. But you can see dramatic survival rescue of the phenotype with iron chelation. And actually, when we take these cells that have had chelation, put them into a mouse, and then track their survival over time, they live for months in the mouse. So it seems like there's a crisis point. It's iron toxicity right then. So if you get the chelator, you get them past that crisis point, and then they might be actually more functional. We could test that in a mouse that has it's a mouse model, a very severe mouse model of PMD, by treating them with a blood-brain barrier penetrant iron chelator called therapeutic. And then we found that the mice had a small but significant increase in survival. But more kind of interestingly, we kind of looked at the white matter. Instead of seeing a lot of apoptotic cells measured by caspase-3, much reduced, instead of seeing no myelin, we did see myelin in the treated mice. So that actually is another promising way forward. And what about fixing the PLP gene? Now, as I mentioned, PMD is caused by duplication. So if you want to do a gene therapy and you said, well, we're just going to do a gain of function. No, it won't work because too much is bad. It's also, if you get rid of all of it, a null mutation is not great. It's a mild form of the disease, but it's still PMD, and it can be caused by point mutations. So it's really kind of tricky. So there's still some things you can do. One would be an antisense nucleotide. These are coming more and more into clinical practice. As you know, we've got DNA, RNA. But if it's making a disease-associated protein, you want to stop this production. So what an antisense does, it can bind to the RNA and can prevent the production of disease-associated protein. That in PMD is predicted to be beneficial. And in this paper from Paul Tazar's lab, here are the untreated chimpanzee mice. They die very kind of quickly. Now put them on the antisense oligonucleotide, and now you have this really excellent survival. And so that actually is going forward to clinical trial, and that should be opening soon in Europe, in Amsterdam. Now, so the good side is the mice are living long. The sort of bad news is what you've done is you've taken a really bad PLP mutation, you reduce the levels of protein, and now you've converted that into null. So now this is now a mouse that is much better, but it actually still has PLP. So the antisense oligonucleotides can't be the final answer for this disease. It could be a temporary, and then it'll be very positive. I think that clinical trial will be really interesting to follow. So what you really want to do is to fix the gene, just like we did in the iPS cells. If you can fix the gene, that cell is it's a normal oligodendrocyte. So could we start to think about CRISPR approaches or base editing approaches? And here are some of our collaborators that are thinking about ways to gene correct the oligodendrocyte. And so we're also pursuing that in the lab. So we'll test it in that mouse, so we can make that mouse better, test it in human iPS cells, and think about whether potentially something we would want to translate. Now, the diagnosis of PMD, it's typically been pretty late because you would need MRI to diagnose PMD. And as I told you, you don't really see MRI evidence of myelin until you're 2 years of age. And that might be too late to kind of have an early, to have the full therapeutic effect. But this is all sort of changing. So here's a case from our PICU at Addenbrooke's, 8-month-old admitted, is floppy, developmental delay, has something called nystagmus, funny eye movements, stridor, so maybe is needing respiratory help, normal MRI. But then here is the whole genome sequencing. We did this from the mom, the dad, the child. The mom is a carrier. She has 100 base pair deletion of PLP, and that's been passed on to the son. So again, this is X-linked recessive, so mom doesn't have symptoms, but she's a carrier. And so that's just an example of how we're going to be diagnosing PMD much earlier, and in general for all the rare diseases. You can look forward to them being diagnosed much sooner. Potentially, that's a more therapeutic window for these two new types of therapies. So again, I think pediatrics is going to start to get these very exciting opportunities to think about, impact of precision medicine on patients that might show more benefit. So here are some of the, approaches that are being, now developed for PMD. The cell-based approaches, iron chelation, also a clinical trial that should start soon. The antisense oligonucleotides is one gene therapy. My lab will be working on the CRISPR-based gene therapy. All of them theoretically can be, more impactful because if you are able to get a few good cells in that brain, expect it to proliferate, migrate, and expand over time, so that you can see, from a single intervention, maybe progressive benefits over time. So they're really cool cells to think about for proof of concept for gene therapies or cell-based therapies. So at this point, I think, I don't know, Jenny, I could finish by 6 o'clock if that's okay. And then just sort of come back to this topic of cerebral palsy, and maybe provide a new sort of perspective on that. So, again, you probably heard of cerebral palsy. It's a lifelong non-progressive condition, affects body movement, causes spasticity or muscle tone is affected, and can be associated with cognitive impairment. Increasing prevalence in developed countries, and this is because of peri-viable infants, like resuscitation at 22 weeks. I don't think it's a good idea, and the child will have probably a greater than 90% chance of having cerebral palsy. So should we be doing that is a good question, but so we're seeing more kids with cerebral palsy over time, and they affect large numbers. The theory for what's causing it is going back to William John Little, brain damage to the brain either before birth or at the time of birth. So now, what about other causes of cerebral palsy? Now, the types of brain injury that you see in babies look really terrible. So you can have a preterm baby. Has anyone done NICU? Probably. I don't want to block NICU, but there's some sick babies. So they can have bleeding in their brain called intraventricular hemorrhage. There can also be lack of oxygen types of injuries. This is the baby that's had bleeding in the brain, which has actually caused damage to the white matter, over time you get hypomyelination, sort of brain atrophy, just because that blood has caused so much inflammatory damage. In a full-term infant, you can have hypoxic ischemic encephalopathy, where both gray and white matter areas are affected. That would be due to some sort of lack of, we say lack of oxygen, but it's actually they're born asystolic, so they have no blood flow. They need to be resuscitated so that they can actually have some perfusion. But that will cause local hypoxic ischemic damage. There's also stroke in babies, and people don't kind of understand. Large vessel stroke is more common in babies than it is in adults. For adults, like we're really good at having small vessel stroke and getting dementia, embarrassing ourselves. But for babies, but large vessel stroke incidence is about 1 in 5,000 in adults. It's about 1 in 2,800 in babies. And that's a stroke. That's a stroking out a hemisphere or a large territory. And that's what you'll see in a baby. They could present with seizures, they could present with hemiparesis, and then by MRI you can diagnose. So those are types of injuries that you, those are brain injuries, not genetic. We can kind of look at what's going on in terms of their white matter and dendrocytes, because now, in addition to O2, we've got other markers to be able to measure their early stage, pre-myelinating, myelinating. We can kind of challenge the prevailing theory. Actually, the theory for why there was lack of myelination in babies with neonatal brain injury was that the oligodendrocytes were super sensitive to oxygen, that they all died. It was based on cell culture data. And then, when you try to replicate it, we found something very different, that there actually were plenty of the precursor cells. But it's just that thing that they got to that stress point and they just stopped differentiating. So there was a lack of myelin. So you didn't have this final cell bind, but the cause was totally different. The cause was that there was a block. And when you think about it in those terms now, you can start to think about what's blocking, can you relieve that block? Is there an environmental factor or pathway that's involved? The pathway that we found that was involved is the Wnt pathway, which again, all singular have heard about, and it's a developmental pathway that's involved in cancer also. Colon cancers, most of them caused by the pathway hyperactive mutations. So when the pathway is active, you can use AXIN2, which is the transcriptional target of Wnt pathway, to kind of look for tissues that have activation of the pathway. Then it's also kind of curious with this pathway. It turns itself off. So you make AXIN2, you turn the pathway on, then that AXIN2 becomes part of a destruction complex that can turn itself off. So it's definitely a pathway that tries to control itself. Things that go wrong in colon cancer is you have a mutation of AXIN2, you can't turn it off. That's sick. Or you have hyperstable continuum that cannot be degraded, and that causes colon cancer. So that's okay. Oh yeah, so this emoji wants us to just keep in mind beta-catenin, because come back in a second. But again, we can check for this idea that maybe pathways turned on in neonatal white matter injury, blocking oligos. We can do that by taking pathological samples from human infants that have preterm or hypoxic brain injury. And what we find is they're expressing AXIN2, which says the Wnt pathway is turned on in these cases, and it does help us to give an explanation for why there is a block. So just that's a little bit of basic science to say that for the neonatal white matter injuries, maturation arrest is a better way of characterizing the phenotype of hypomyelination and cell death, that we think the activation of Wnt pathway is playing a role in kind of blocking them. And we can find this in pathological samples from humans, including those hypoxic brain injury, and also we had one kid who was a 12-year-old who died of cerebral palsy and had bizarrely oligodendrocytes very, very active for Wnt pathway targets like AXIN2. And it really looked like a great sort of confirmation of this mechanism. Now, what will you do about it? It's sort of say, well, maybe you want to inhibit the pathway Again, we can come back to whether that should be additional. Okay, let's get back to William Little. Here's our case. You have this baby who had hypoxic brain injury, and we're asking the question, will you benefit from genome diagnosis? But I told you, cerebral palsy is a non-progressive condition, and let's look at some of the background data too. So now studies are starting to come out from Canada, from Australia, saying people with cerebral palsy have a 20 to 40% rate of a monogenic condition. So actually, their spasticity has not been caused probably by brain injury, but it was something that was caused by a genetic condition. When we looked at our next generation children's project cohort, we had 15 kids with cerebral palsy, 4 had monogenic condition, and that gives us a cerebral palsy hit rate of 27%. Also, when we looked at other cases of, in our series, we did 20 kids with hypoxic encephalopathy. They didn't have, well, 1 in 20 had a genetic condition. So we wouldn't necessarily do whole genome sequencing for a kid with hypoxic encephalopathy, but a child who might show signs of cerebral palsy at 18 months of age, should you be thinking about a genome diagnosis? And let's think about this kid. So in contrast to what I told you about a non-progressive condition, this is progression. He's getting worse over time. So now we might say probably. This is starting to fit the pattern of the genetic form of cerebral palsy. So we're just starting to think about cerebral palsy in different ways. And this is a condition where it's a very sort of somewhat controversial and charged because it's highly litigious. It is, cerebral palsy is the leading cause of litigation in the NHS. There's 37 billion pending litigation for cerebral palsy. The obstetricians are terrified of this, and the lawyers might be think it's kind of cool because it gives them a lot of work. So you get these different definitions of cerebral palsy, and there's a lot of, again, terminological, debate. These sort of things where we should call it this, you should call it that. Is it cerebral palsy pejorative? We should call it spasticity, and we spend a lot of time on terminology. So this is like doctors really argue about how we should call things without really knowing what to do about it. Lawyers give you a different view because they want you to come to them and give them some cases to work on. There are also companies that say, oh, well, we've got to build the cord blood and we'll give it to your kid with cerebral palsy, and maybe it'll help. And we'll charge you 30,000 pounds. And parents are desperate and they're being taken advantage of. Of course, it doesn't work, and there's no evidence that it works. And so it's like it's a really hard for families to navigate this. And I think now what we're looking at is another thing to think about, which I can't say is going to be straightforward. What if there are a significant number of kids with cerebral palsy that have genetic conditions? Okay, well, how good was the history for brain injury? Was there a history of it in Europe? Sometimes moms say, for 2 days, the baby stopped kicking and then it kind of got back. That could suggest there could have been like a transient cord knot and lack of oxygen. So there are in utero events that could happen, placental abruption or other problems. So was there that history? The answer is no. Are the symptoms progressive? The answer is yes. Well, maybe you should think about whole genome sequencing. That's the type of way that we're thinking about this. Now, the way we're testing it in the UK is we have a study that's ongoing now in Cambridge and Colchester and Newcastle, where families that go to cerebral palsy clinics are offered whole genome sequencing, give them full, tell them about the risk, but you tell them if you're considering litigation, you may not want to know the answer to this because if you have a genetic form of cerebral palsy, would you be able to get a settlement? Or you can actually start to see how if 1/3 of the cases of cerebral palsy are due to genetics, then the NHS may, they won't pay those claims. They won't be able to prosecute a cerebral palsy case. And so it's going to really have implications around all of that. But then you could also say, on the other hand, well, maybe we'll get some answers that would be helpful for that child. And I think this is where Clare is sort of taking the lead and saying there are genetic forms of cerebral palsy, and now we're using that to stratify our approach. because you might be getting baclofen or rhizotomy, or there may be drugs that be helpful, or there may be gene therapy. So there's definitely an upside as we start to think about taking this nebulous term cerebral palsy, because no one can really agree what it means, and now starting to parse out and stratify patients to sort of see, will they be eligible for clinical trials? That's the way we would like to take it forward. But again, it just shows you like there are a number of controversies when it comes to genomic medicine you have to consider. And then there's finally this thing that we noticed in one paper, which was genetic forms of cerebral palsy, and the beta-catenin was coming up. And actually, it's one of the most common variants that shows up in cerebral palsy are mutations in beta-catenin. And there are 67 reported patients with cerebral palsy, and they occur in different parts of the protein, but some are activating mutations of beta-catenin. And then you'd say, oh, well, David just told me that activation will affect the oligodendrocytes and cause them to not myelinate. So can we prove that mechanism? So that's another direction that this field is going. We're going to be getting better at diagnosis. We're going to also get these questions, and you say, oh, we'd like to take that to the lab, and let's model this. And we're doing this now in iPS cells, and also with Melissa Gammons, the Cambridge Institute for Medical Research. There'll probably be a PhD student in there somewhere. But the idea is now we can take patients or beta-catenin mutations, put them in oligodendrocytes, and see if those oligos are blocked from myelination. Put them in a mouse, and they can start to now see if this Wnt pathway makes sense as an explanation for these patients' variants in cerebral palsy. Now, that would then say we have an insight. Now we can think about precision medicine. And, if we follow the outcomes of our intervention, obviously, we can get a better sense of how that's working. Again, this is the direction of travel. This is where pediatrics is going. And when we think about things for the brain, we have to think about precision delivery and also the nature of gene therapies. Here's one of our pediatric neurologists, Pooja, who's leading a new gene therapy trial for Angelman syndrome, which is an antisensitive study. So that's happening here. And as we go forward, we want to be able to do this with more precision using intraoperative MRI. This would be with neurosurgeons who would now be using special catheters and design ways to put things into the brain, target special cells or regions in the brain, in as safe manner as possible. And that will hopefully be a new unit that will be opening up next year in the clinical research facility, which is on the Paediatrics campus. So we're calling this the Advanced Therapy Center of Excellence for Neurological Disease, or ASCEND, and that will be also networked with Queen Square Neurological Institute in London. And it could actually be part of an international effort to start to think about precise, safe delivery of brain therapy. So the rare disease diagnostic odyssey is kind of shown here. I think it's kind of not going to be relevant in the UK because we have now whole genome is really becoming much more available. The newborn genomes program is really taking it to the next level, almost I would say too soon. Whole genome sequence screen for newborns when they're born, such that somehow we'll be able to predict something about their future, which unfortunately we're very bad at doing. So we're going to have the test before we really know how to use the test. And I'm sure there's some ethicists that's screaming about this, but GEL is doing it. And so more and more, I think pediatrics is going to need to be able to cope with this type of information, and parents and health systems are going to expect it. And this is where it can happen. The new Children's Hospital will have genomic medicine center, neuroscience, the types of things like Pooja is doing with gene therapies. This now is sort of moving forward on the Adams campus, across from the Rosie Hospital. So that will be something to look forward to in terms of these new therapies. So this is just a summary of what I've taken you through, which I think in the interest of time, thank you for your attention. See if you have any questions. Sorry if I went a little bit too long. Too small group to clap. I think this is it. We'll give it
I have a question about there would be implementation of newborn screenings especially how is
well, it's almost a future question, Jenny. So the way that we did our study, we started from an ICU, and the kind of proposition that the whole genome sequence, there may be a diagnosis of helping the child. Parents are highly motivated. Everyone wants to know the answer quickly, and we have a team that's able to support them with the diagnosis and then change the therapy. In some cases, sadly, the change in therapy is far place because they have fatal condition. And you could argue that's also kind of humane because instead of having to solve one, that's a problem much longer trying to figure out some kind of but then we also have a PhD student in psychology who's going back several years later to the same family and looking at the psychosocial impact of having had it diagnosed when they were a baby. To sort of see, if they have been supported, would they have rather not known? Hopeful, you don't know. And so we're actually trying to check how our approach landed with families over time. And that would be an example of, I think, what we should do is sort of, check back. And there is, it is new. There's a lot of uncertainty. We were looking at kids in ICU, so the question is, could we get diagnosed the condition that goes into ICU? The newborn screen pilot, genomics in the limited newborn genome program, I think it's like a harness because you're going to be doing this on healthy babies, and then you may say the baby has a 25% chance of getting prostate cancer. It'd be like 23. I mean, it drives people crazy. There are all these risks and there are these uncertain risks, imprecisely. And some of you could say, like the counter example, you have Huntington, you have a 100% sensitivity. That's an example of a lot. And sadly, so things can't die. That's what we want to do every single day. Is that you have a 100% chance of getting it. Therefore, you're going to prevention. But to do that, we need to develop these types of databases where we have genes and outcomes. We need to be able to look at the environment, school performance, the health record over time, develop couples. Also, all the latest standards, the genome, transcriptome, the proteome, the microbiome, and actually the columns predict the tool that we in the future, taking the kind of complex factors, and then we can get virus for learning. Therefore, we get some sort of program to prevent that out. And then we say ahead of the negative or cost, you can have both. 35% chance so we have another test. So you have to do more and that's organization and parents, they saw she got better with change medication. That's how you want to be committed to it. Unfortunately, the field is actually causing the head, and so I think it's risk that there's a backup.
Thank you. So my question is about epigenetics and if there's tools or develop genetics in the genome changes the gene.
Yes.
On the level of epigenetic changes that make, is there any work that has been done, and is there any progress for
some that are being developed for cancer. The person who's very keen to do that genetics actually would be Matt Hurls, who's the director of the Sanger, and Anne Ferguson-Smith, who's the Pro-Vice Chancellor for Research in the University of Cambridge. So your questions are like a very high level. You know, the proposal that we're putting forward now is to say, we want to say that we need to learn more about this genome and the outcome in a really sort of a well-designed birth cohort study. Baby's born, we'll have a genome, but we're also going to do methylome to address epigenetics. Transcriptome, we're going to look at single-cell transcriptomics of T cells to look at certain inflammatory markers. Proteome, because there are some things that would just show up as a protein deficiency that you might not have predicted from those other things, etc. And then in the short term, we would say, can we improve our diagnostic rate by having this multi-omic approach from, let's say, 25% diagnosis in NICU to 30%. And then long term, do some of these markers mean something? You start to connect them to health record, educational record, you know, sort of other trajectories that you can measure through electronic record linkages, which we're doing in something called the bioresource, which is something like UK Biobank. And it's a system we're just sort of putting up. So we are proposing to test that. And then the proof will be, I guess, when we put it all together. Question. Yeah. Yeah. They have one in Stanford too, because when UCSF had one, they had a copy. Yeah. No, it's really different. It's really kind of, you know, it's a very different landscape. So, you know, for the neuro-NICU, there's actually no evidence that it's better care, because, you know, often, because it's hard to kind of compare one thing to another, a lot of changes in care. So there's this feeling that it's better because you've got all these measures, got special nurses, special doctors. You know, I mean, when you're a clinician practicing, you actually can sort of see improvements in care that you know are better. But if I was supposed to say, did the kids get into college or something, there's no data like that. But in the US, it's all about competition. And so the fact that UCSF has it meant we were getting all the patients. So then Stanford said, oh, we have to have one of these things. And then Kaiser, which is another Bay Area. So now the Bay Area has 4 neuro-NICUs, you know, and then, you know, parts of the country, many cities in the US will have 0. It's just like this competition thing. For genomes, we can get genomes here because the government and now the new foreign secretary, you know, put into place when he was prime minister, the 100,000 genomes project. You know, David Cameron had a son with one of those rare epilepsy syndromes, and that's probably why he, you know, felt this would be a good thing for the UK to push. It was, you know, I don't think he did everything right, but I do think that was a good call, and it really did sort of make the UK a champion of whole genomes versus exomes. Big debate in other places, and also we're way ahead of it at pretty much any other country at the scale of the NHS, which is 66 million people. So there's just big cohorts here. And the US, it's like, does it save money somehow? So now you have to take a genome, you have to say, and then the argument is really like kind of a bit depressing. So the people that are pushing this, there's one sort of evangelical genome person who's in Northern Irish called Stephen Kingsmore at Rady Children's Hospital in San Diego. And there's some people that really exaggerate the benefits of the genome. And so they have a study that said, well, we did this genome sequencing and we saved $2.5 million on some metric. And but actually what they did was they made fatal diagnoses through life support. And so it was the savings of hospital days on life support, which in the US are very, very expensive. So a baby on a ventilator a sort of high level, let's say dopamine, like a very sick kid. In the US, that would probably cost about $50,000 per day. That's what the hospital charges. So if you have less hospital, but it's not a very nice message. I don't think that's the right way to make the case that whole genome is, but that's kind of what you have to do. So he's now got some funding in California to pay for genome based on terminating life support argument. You'd much rather see like the case of that baby I said where they were in status, then they went on carbamazepine, woke up, started breastfeeding, home with mom 3 days later. That's the story you want to shoot for and show those outcomes. But in the US, that's actually what would be needed. Or it could be like you just go into this competition and say, and I just noticed UCSF just did this. They said we're the only hospital in Northern California that's offering whole genome diagnosis. Now Stanford's going to do it. I think that's because now it's like this competition. But slowly but surely it's going to catch on. These numbers are you can walk away from these numbers. Pediatric neurology 1 and 2. Probably in rheumatology, there'll be a number of other specialties. Pediatrics, probably pediatric subspecialties or repeat hospitalization will be an indicator of a probable genetic condition. So and when it becomes essential for diagnosis, somebody's sued because they haven't been made the diagnosis when they could have. That's when it'll change, and that's going to happen. I think the next couple of years.